colleagues. Good evening, good afternoon, good morning. From wherever you are watching us tonight, welcome to our webinar where we will be discussing the difference between biologicals and biosimilars from the regulatory perspective. Please feel free to write in the chat box and let us know more about you. What do you work? Where do you work? Are you working in the field of R&D, regulatory affairs, or are you a student? Please let us know more about you by writing your position in the chat box. Also, if you have any suggestions, please write them in the chat box, which will be open for the beginning of this webinar. Also, if you have any other suggestions on any future topics that you would like to have a webinar or attend a webinar on in the future, please write your suggestions in the chat box. We will start our presentation in a few minutes. After a few minutes, the chat box will be closed. So please, if you have any question during the presentation, feel free to write them in the Q&A section only. We will be taking all the questions from there. Okay. As you all know, we live in the era of biological medications. Biologicals are globally used for a wide range of diseases, including serious and life-threatening conditions such as cancers and rheumatoid arthritis. We will also be discussing biosimilars, the medications that had brought competition to the market, expanding patient access to novel, affordable, life-saving medications. Regulations are the only way to guarantee that only safe, effective, affordable medications with very high quality are provided to support all patients around the globe. We would like to take the opportunity to thank our outstanding guests for joining us tonight, facilitating knowledge transfer for healthcare professionals who had joined us to hear more about such an important topic. It's our pleasure and honor to introduce our expert, Dr. Hakima Hose. Dr. Hakima holds a bachelor degree in pharmacy from the University of Jordan with more than 30 years of experience in the field of regulatory affairs. Also, she's a regulatory affairs certification holder. RAC is the leading post-academic professional credential for regulatory affairs professionals who are also a member of the regulatory affairs professionals community in the United States. Dr. Hakima has a unique experience since she was the head of regulatory affairs department at the Jordanian FDA. Then she moved to work at Hikma Pharmaceuticals as a regulatory affairs and due diligence senior manager. So today we will be hearing from two perspectives, the regulatory authorities and the manufacturers. During her long experience, she had drafted many guidelines with JFDA, such as technology transfer guidelines, biosimilar registration, cosmetics, radiopharmaceutical stability, bioequivalence, in addition to the value-added medicine guidelines. We would like to thank you all for joining us tonight, and I wish you a very nice, informative evening. Now, I will leave the floor to Dr. Hakima to start our presentation for tonight. Dr. Hakima, the floor is yours. You are more welcome.
Thank you very much. Assalamu alaikum wa rahmatullahi wa barakatuh. بسم الله الرحمن الرحيم والصلاة والسلام على سيدنا محمد أشرف المرسلين. As a start, I would like to thank VBN for giving me such an opportunity to join and to talk about such really very big and huge topic, and really it's the start of art of the now that everybody really concerning about the biologicals and biosimilars. I hope I will really make the best of the time to have really a type of consolidated regulatory perspective to understand biologicals and biosimilars. As I mentioned, the topic really needs 2 days or more. I tried my best to summarize it in one session, 1 hour session, which it will be really, it is a difficult thing. I hope I have succeeded. The second thing I would like to have really to listen to you and to have your questions, because this will guide me how to answer you in proper way. Let's start, and Bismillah. Thank you all for joining, and really it's a pleasure for me. Thank you.
Dr. Hakima, I want to highlight for our colleagues that the chat box will be closed now. Now my colleague Rafa will add the registration link, so you can join our IVBN Regulatory Affairs List serve and be a member of IVBN Regulatory. We will be pleased to have you all. Please, during the presentation, if you have any question, write it in the Q&A section only, so we can answer all your questions. Wish you a great evening. Dr. Hakima, the floor is yours. Thank you.
Thank you. Thank you very much. Bismillah. So just this presentation is totally representing my own thoughts and my own experience, not Hikma thoughts, of course. In this presentation, we will try to have a little bit about knowledge about biological product, biosimilar product, differences in registration requirement, guidelines all over globally, complexity of registration process, why we have really minimum number of biosimilars compared to generics products, for example. What is the similarity, how to interchangeability, immunogenicity, and all these and much more in this presentation. Why pharmaceutical industry shifted to biological products? Really, this questions have been raised since 1980s, where industry have going toward biological products very instantly and focusedly. The thing that biological products act as focused stratified approach. I mean that the product, it works not as general. It will not be distributed in all the body and work there. It is really localized and have a focused medicine. So it is customized to the right patient and to the right therapy. This is really the aim of biological products. This is why, and it was really the hope for everybody in order to reduce the side effects, to reduce many things, because we don't want the whole body to be involved in the treatment. So to focus as much as we can and to customize to the right organ and the right disease that we need. That's why really this approach have been growing very fast, and everybody is really looking forward such biological products. But nevertheless, this was really a very long debate about the introduction and how we will go there. And of course, we are talking here about biologicals, not biosimilars, because biosimilars lately introduced to the market. The questions were, what is the real benefit of them? And how will this help a patient on long-term condition? What will these new therapies mean to the industry? But as we mentioned, it is really going very fast, and now really by 2020 it was more than quarter of the entire pharmaceutical markets. So the biological products, if we want to define it, it is a biological products contain components from living organisms. So they are living, such as human, plants, animals, microorganisms. They have a more complex structure, larger molecules, and are not easily reproduced. So from batch to batch, there might be variations. Specifically, most biological medicines are developed to create the required proteins rather than through a traditional chemical synthesis. They are produced by genetically engineering living cells using recombinant DNA. The technology approved based on full dossier, and in most cases, it is the reference product. This product is approved according to a full dossier. We are talking about full clinical studies, full quality, full non-clinical content. Examples of biological products, and which include vaccines, blood and blood products, gene therapies, tissue and transplant, recombinant proteins, stem cell therapies, monoclonal antibodies, and all these are examples and which are used now, very much used. These are the top selling products in 2017. Humira, you know, everybody, Isla, Revlimid, Rituxan, Enbrel, Herceptin, all Avastin, Remicade. These are one of the that it is really used and very much used. And now by 2022, the numbers are much more, and they are have the biggest market share in the market. A biosimilar product, which was introduced late, as I mentioned, a biosimilar is a biological that is highly similar to and has no clinically meaningful differences. I have highlighted these two words because these we will concentrate in our talk today in order to emphasize how we will prove these similarity and no clinical meaningful differences from another reference biological product. Biosimilars bring competition. Really, they have been reducing the prices, reducing increasing the availability of the product, so more patients can be treated for less money. But still, we have very slow uptake of these biosimilars. Why? The answer is very simple because of regulatory hurdles and vague guidelines of biosimilars. Really, there is a type of, let's say, a type of worries always, and a type of fear of approving the biosimilar products due to such complexity of the molecules. Highly similarity or no clinically meaningful differences between it and the original biological products, really this is very difficult to prove. Just a comparison between the chemical product and the biological product. If we go for the size, you know, the chemical products is very small, while we have a very complex, large proteins, peptides, nucleic acid cells of the biological products. The example is that this is the structure molecule of aspirin, while this is the insulin. This is insulin and this is aspirin. You can see how much the differences. A method of manufacturing, very easy to produce, stable, and we can guarantee the production of the product. But here it's very difficult using the biotechnology, very difficult to characterize, sensitive to any changes. The source, as we mentioned, chemically, while here biotechnology using animal or living sources, mammalians, bacteria, yeast. The method of action here is not specific. The chemical product reach the whole body, and it works wherever it needs, and it might even cause side effects. Here it is very specific, lower toxicity, higher safety profile for a biological product. Now, the immunogenicity, which is introducing an immune, this is non-antigenic, while this is very antigenic or might cause very immune actions in the body. Method of administration, orally injectable, you can find any type of dosage form. Right here, it's really, we have a minimum, we have either injectable, inhalation, or infusion. We don't have much more. Now we're starting introducing a new, for example, to focus on the oral, but still, if they are in early stages. The price is cheap while it's an expensive one. Dossier assessment, simple, clear. We have harmonized, almost harmonized guidelines. Not clear, not harmonized all over the world. Interchangeability and substitution. Here we can substitute with any generic product for the originator, and here the substitution of biosimilar. But is this true? Can we interchange and substitute any biological products with a biosimilar? This is the question that still really needs an answer from the authorities, from the manufacturers, from everybody. In general, biosimilar guidelines in EU and USA. Sorry, Bayan, but we will. I will finish the presentation, then we'll have the questions. Yeah, Bayan.
Yes, Dr. Hakima.
Yeah, this is okay. Thank you. Just to make sure everybody is aware of this. Please write your questions anytime, and I can go back for any slide if you wish to have more to explain more or any to clarify more.
Yes, I thought this would be better, so they became focused with the ideas and they know the differences. So I thought it would be better to take the questions at the end.
Okay, great. Agree. Thank you. Perfect. Now, biosimilar guidelines in Europe and USA in general, as I mentioned, no harmonized guidelines, even between Europe and USA. In MENA, usually most of MENA countries adopted EU guidelines, but still assessment and evaluation are ways afar between each authority. So number of approval of biosimilars are still small, and number of locally manufactured are minimum. We will know the reason behind this. European Medicine Agency, really, it was the first to introduce the biosimilars regulatory framework in 2006, and while US have been delayed later, EU have developed specific guidance for each type of biological product. So we can go for insulin alone, for MAPS alone, for monoclonal antibodies. Each one has its own guidance. So if you search for guidelines, you just search for the type and you go. While in USA, the trend was in different way. They have really adopted the guidelines started from 2009, and it has been a part of their act in 2010, created abbreviated approval pathway for biological products that are which is a follow-on. Even the naming of in USA was a follow-on biological, not a biosimilar product, while biosimilar is the name from Europe 1, to be highly similar interchangeable. Recently, in February 2022, USA have established guidance for the quality of the dossier, and it's really a very nice one. I have added it to the references if anybody interested in such guidelines. Well, let's talk about our main three countries, Jordan, Saudi, and Egypt. In Jordan, first biosimilar guidelines were issued in 2008 and updated 2015. The first country in the MENA region who have started biosimilar guidelines. GFDA adopted EU regulation mainly and referred to FDA in some articles. In biosimilar guidelines, blood products, vaccines, and plasma-derived were excluded from this guideline. Saudi FDA didn't issue a biosimilar guidelines, but issued a guideline in 2017 regarding the quality consideration, how you prove the quality and how you want to submit a quality module 3 requirement for Saudi FDA. And that, of course, it follows all EU and FDA guidelines for approvals. Biosimilars in Saudi have been given the priority, and the last guidelines issued February 2022 have given biosimilar products a fast track approval. and abbreviated approval. In Egypt, the guidelines were early in 2013 and updated March 2020. Egypt have full, really detailed guidelines. I have been honored to review the guidelines with the Egyptian authority in 2015 and 2013. And we agreed to have, in that we have commented, and really they were very flexible to take our comments through workshop with the Egyptian authority. There they have adopted two approaches: standalone approach and biosimilar approach. What does this mean? Standalone approach is that any applicant can submit a biosimilar product, not a biosimilar. I mean, there is a difference between the biological reference product and the new biological product, which is in somewhat biosimilar. But the applicant have submitted all the dossier, the clinical part, the quality part, all modules. So there are differences in early stages in formulation or in the formula. So he rather to go to standalone approach and to be approved as a standalone approach. So it's not anymore biosimilar, but it is a standalone product. Biosimilar approach, which you have the same and you have all the comparability studies. Blood and plasma drug were not excluded from the guidelines. The guidance includes details of submission process of imported products from reference and non-reference. In Egypt, there is differentiated between reference country and non-reference country. So they have two methods of approval. And the timelines are different. And then if you are from non-reference country, you need to submit for exemption from box, from approval, and then you'll go for the whole process and assessment. But if it is from reference country, you cut these two steps before box submission. Box approval applies for biosimilar products like the generic product, but I don't know if everybody knows about box, but please, if you don't know, write it in the question. I can give more details about this, but they have more chance of approval than, of imported biosimilars more than the imported generic product.
Doctor, can we give a small hint on box approval in a very quick, in a brief way?
Okay. In Egypt, number of permitted products per any product are 12. One for the originator, which is, we call it a box that have 12 products. One, the originator. Now I'm talking about chemicals. One for originator, one for imported generic product, and 10 for locally manufactured products. Okay, so you need to submit to the box first, and then you have approval in order to proceed with the submission and to if you get an approval. For pricing step, the second is start pricing step, the second later, but after it is the technical approval. Now, if for the biosimilar products, they give a chance for 4 imported biosimilars more than the generic products, and less for the locally manufactured, and of course, and if we have 1 reference product. Now there is a degree, in recent degree, you can be exempted from the box if you prove the need. And this is really a new process that you can submit for exemption from the box approval. And you can pay for more for this product, and you can proceed with submission. But nevertheless, this is especially for Egypt. Some countries like Lebanon have such type of box, but it's not in the same way as Egypt, but they have a limited number of imported generics. As Algeria have, of course, also the same type of protection in a way that they, for example, if you have a locally manufactured product, they banned importation. But the type of box is really typical for Egyptian authority, and other countries have type of protection, but in different way. I hope I have briefing it in a good way.
Yes, thank you.
Details of locally manufactured product, they permit to be locally manufactured, and you have to submit for scientific advice. Really, for scientific advice, is implemented and it's adopted by all the authorities, FDA, Saudi FDA, Egyptian, and all, and of course European and the JFDA. And this is the most important way to prove the biological product or biosimilar products, to go for a scientific advice before starting, because the cost of development, the cost of production, the cost of clinical, the cost of quality is really huge. So in order to make sure that you are going on the right track of approval, you need to go for scientific advice. So for locally manufactured product, you can submit for the formula and you wait the answer from the authority, and then you go step by step how to prove. And we will go through this stepwise approach later. So if we go, we have said that it is the difference between chemical and biological product. Now, what is the difference between the biosimilar and generic product from also regulatory approvals? And biosimilars are not generic. This is really, we need to have it in our mind. It's not a generic product. Generic drugs have the identical active ingredients to the brand product and must demonstrate bioequivalence. This is the most important thing. You want to prove a bioequivalence study through fed and fasting bioequivalence study and all this stuff, and you prove efficacy of the product and you are similar to the originator product. While for a biosimilar, this is not the case. Bioequivalence studies will not work for a biosimilar product. Must demonstrate high similarity to the reference product, and there is no clinically meaningful differences regarding safety and potency of the product. It is very difficult to characterize and analyze from the quality attributes of the biosimilar products. We are talking about we are talking about a product introduced into living material and cells. We have cell banks, we have master cell bank, we have tissues, we are talking about proteins, peptides. So so it's very difficult even to characterize and to analyze. The way I usually describe a biosimilar product is how we change through, for example, temperature or through boiling or through the egg. The egg is, it's usually normal, it's liquid, it's protected. And once you put it in heating, you will end up with another solid material, different structure, different color, different from the original one. This is the way that I'm always imagine the biosimilar product upon production and the process, manufacturing process, and the biotechnology. Highly dependent on the manufacturing process. Please keep this in mind. Usually we mention the product is the process. The biological product is the process. This is really a wording that always you hear it. The product is the process, because the manufacturing process of a biological product is very, very important to determine product quality, safety, and efficacy. because any changes, any minor changes, even very, very minimum changes will end up with different either immune response, different efficacy profile, different impurities, different, you will end up with a different product. So it is highly dependent on manufacturing process. So keep this also back in your mind. So once we say that we need a highly similar product, no clinically meaningful differences. What do we mean by that? How we can really achieve such similarity and such no clinical meaningful differences? Once we say a highly similar, we are talking about same weight, same strength, same dosage form, same everything is the same as the reference product. Therefore, can be taken the same way by the original biological medication. So nothing changed. There is no type of what's called hybrid or value-added medicine or interchangeability between capsule and tablet. That's what we would talk about once we talk about a chemical product. You can replace a tablet with capsule easily, and you can use it, or with suspension, for example, if you can just adjust the dose. But when we're talking about biosimilars, this is not accepted at all. Highly similar, so it means highly similar to from every aspect of the product. So what do we mean by no clinically meaningful differences? You have no any clinical meaningful differences in safety and efficacy. So we are not talking about just efficacy, we are talking about safety compared to reference product. The same high standards of manufacturing also. So once you are talking about the manufacturing, the way of manufacturing, the cells, the millions that we started with, whatever we have started, we need to go through this process, and it should be the same exactly with the same high standards. It's not permitted to have, for example, some impurities ranges or whatever the purity to range from that to that. It should be the same. So this is the most really challenging efforts that we need to go in order to prove a biosimilar product. And this is the challenge for also the authorities to and how they will make assessment of the dossier to prove the highly similarity and clinically meaningful differences. So this is the question that everybody really raised. Either we are talking about authority, manufacturer, even the physician who will use this product, the patient to trust to use this, how to prove the the similars. When we are talking about a biosimilar dossier, we need to have these concepts to consider them. Since, as we mentioned, biosimilar is not a generic product, so approval approach of generic is scientifically is not applicable. So I will not go ask for fit and fast bioequivalence study in order to prove a biosimilar product. So I will go for different studies. Second, characterization of generics don't apply to biosimilar. More characteristics exercises of comparability studies. We are talking now about comparability studies, which is really comparison head by head between the reference products and the biological product, are required to prove equality, similarity, efficacy, safety. And as I'm here, as you can see, we are talking about quality, safety, and efficacy. So we are talking about comparability studies regarding quality, efficacy, and safety. We are not talking, for example, if we can prove the quality, then we go just for efficacy. No, we are talking about the three modules. It's due to biological origin and biotechnology. Biomanufacturing is the production of biological from living cells, as we mentioned. So companies use the process to make the biological process such as antibodies and enzyme replacement therapies. So we need to take care of this biomanufacturing process. It is very sensitive. Compatibility similarity exercise should be head to head. I mean, that means that every property of the biosimilar should correspond exactly the same to the reference product. That's what we mean by head to head. So no side to side, by side comparison of a biological product usually apply to the quality. But head to head, we are usually go for the efficacy and the clinical studies. Any approach of submission of the biological products, either the biosimilar or the original, it is a type of stepwise approach. In submission, scientific advice in all steps is recommended. You can't go for any the next step without really making sure that you are the authorities are satisfied and they agree with your approach in approval with the first approach of compatibility studies. So the registration requirement, any biosimilar products considers a new drug. So there is no abbreviated submission applications. There is no generic application. There is no, it is a new, completely a new dossier, a new product considered. Of course, CTD or CTD depends on the authorities' requirement. Biosimilar can rely on certain existing scientific knowledge about the safety, purity, and potency. There is a reliance on the biological product, reference biological product. But how much this reliance, this is what we will see, how much we can rely on that, on the information that has been published for the product, the reference product. Authorities have the right to consider what data should be submitted. This is the most critical thing, because if I'm sure that I have submitted all the data and I'm happy about my product and I'm satisfied about the similarity, the authorities have the right, and this is written in all the guidelines, that to ask for more data, to ask for more studies, clinical studies, or to give a conditional approval and to ask for more studies after approval, post-approval. So they have the right to do that. This is why we need to go for the scientific advice before, in order to have really to be aligned with the authorities' consideration. And even though after we finish our exercise and the compatibility exercises, they might ask for more studies. Authorities evaluate each biosimilar product on a case-specific basis to determine what data are needed. Also, this is very, very critical, because you can't say that I have COVID, for example, an insulin which is the same as that. No, each insulin and each product and each biosimilar product is a case-specific study, and the authorities will go for a case. Why? Because even the starting material, even if you change, for example, the diluent, even if you change the culturing area, while you are doing the cell bank, even if you started with different room temperature, different conditions, different humidity, you will end up with different protein. And that's why we have to consider assessment through a case-specific basis. There is no two typical biosimilar products in exactly the same, even though they are the same product. At the end, we will end up with the same product. It has been really the challenge that the authorities, for example, FDA authority, US FDA, have an approach of categorizing antibodies and number of the amino acids in each protein in a different way. So they have three categories. Those less than 40 amino acids, 40 to 100 amino acid, more than 100 amino acid. And there was an approach that to consider those less than 40 amino acid or 40 to 100 as a type of, let's say, simple biosimilar products or a simple biological product, so they can have been approved in a simpler way than the usual biosimilars approval way. But really, this have been for a while, for 2 to 3 years, and at that time, insulin was considered as a chemical, can be assessed and be approved as a chemical product, not as a biological product. But now, of course, FDA have revised this regulation, and now insulin, one of the biological products, with minimum, with a less requirement, for example, regarding immunogenicity, is not anymore required for insulin product in comparison with European guidelines or other guidelines. So that's why we are talking about a case, a case specific basis of to determine the needed studies for each product in order, and what we can waive or not we can waive of study. In this case, for example, if you can improve similarity inequality, and especially for the purity of the cell bank and the master cell bank, and there is no impurities, this might minimize, for example, the safety studies, compatibility studies. So if we prove original equality, very, very similar equality, we might have less studies of, for example, non-clinical studies, or less of studies of clinical studies. So that's why we need to go stepwise approach in order to make sure what the extent of the studies that we need to go, and how much we can waive of these studies. All comparisons should be head-to-head, as we mentioned, using a state-of-art method of analysis. This is very important, because if we go back to, for example, for our old method, this will not be acceptable by any authority. So we have always to have the state-of-art method of analysis. If we started with a reference products, we should continue all our study by the same reference product. And it should be one reference, inequality, in non-clinical and clinical studies. We can't check, for example, even the purchasing from different countries is not permitted. You have to start to the same reference all through the studies. Compatibility studies should be demonstrated from drug substances, and we start as early from the master cell bank with comparability studies. It's not once we finish up to the finished product. No, start early from the cell bank until the finished product in order to have to prove similarity. If differences appear in quality, then here this is really very important, and applicant or manufacturer should consider a standalone dossier. And in such a case, he will not go for comparability studies. He will do all the studies as if it is a biological product. So if differences appear starting early from the quality, go for a standalone dossier. And also here, and you need to justify, of course, very scientific justification why do you have such differences, and in order for the authorities to accept the standalone dossier. Jordan have accepted that, and it's mentioned in the guidelines. Saudi also, Egypt also, Moroccan also. Although in Europe, there is nothing mentioned clearly about standalone dossier, but this is really permitted in a way if it is justified scientifically. Extrapolation of indication is not accepted by all authorities. But now, unless justified, of course, but now some authorities, as the US FDA, have accepted extrapolation if biosimilar may be approved for an indication without direct studies, without head-to-head studies of an indication, if the total evidence in the biosimilar application support a demonstration of biosimilarity. So this is very important, really, let's say, upgrading and updating in guidelines, because in early stages, once we developed the drafted guidelines in 2008 in Jordan, it was really not permitted at all and not accepted to extrapolate the indication. And each indication need a head-to-head study, clinical study, in order to prove. But this updating, now it's really relaxing a little bit the manufacturer and the guidelines and the requirement if we can prove total evidence of biosimilars. We'll talk about what is the total evidence of biosimilarity in a way. It's a must to be able to trace and to track and distinguish between biosimilar product and difference. Why? Because if you started your patient on a biosimilar product, for example, and if you shift to the biological product, then this will mean that if any immune or immunogenicity have established in the patient, we will not be able to distinguish between from where this really immune response have developed. Is it from the biosimilar or from the biological product? So it's very important to be able to track your biosimilar through naming, batch numbering, traceability, tracking, and patient name, physician name, hospital, date of submission, the way when it was submitted, in order to recognize if any immune response have developed from this product. A risk management plan, it's a part of the dossier. You can't submit any dossier without a full risk management plan, pharmacovigilance plan, and post-marketing follow-up clinical studies. Usually, by most authorities, you have to follow up your clinical study, your product, either as immunogenicity studies or as a clinical study to prove, until it is proven that there is no difference between the biological and biosimilar products. In naming, usually we develop WHO guidelines. FDA have, US FDA have a new system to add the first name of the biological product or the annex of a biological product in order to refer to the the biological name, and of course in addition to the company manufacturer, in order to be able also to track and to trace the product. Approval process of biological products and biosimilars. Usually, either it is a sequential or parallel starting from the site. It usually it is a simple dossier, same dossier as the CTD, full CTD, ECTD dossier, with full studies of each one. But we are talking about patients, clinical studies in patients. We are talking about a biological product. So we started with cell bank, with the master cell bank. The site to be approved, it's starting from the early cell bank studies, not from the, for example, finished product or like chemical products. So we have to start early with the sites, and then we'll go and the dossier. Either it is a parallel or sequential, depends on the authority way of approval. Usually, scientific advices are very important in any biological application, and it is part of the dossier. We need to have to include all scientific advices and result of scientific advice and meeting with the authorities in the dossiers, because this is really our guidance. Although it is not, it's not binding for the authority, although it is a scientific result from them, but keep it in your mind, it's not binding. I mean, because the time of developing a biological product and the time of approval might take 2, 3, 5, 8 years. So by that time, new things might arise, new state-of-art methods, and new knowledge, and new safety about safety and efficacy. So even scientific advice, although it's a stepwise approach, and we have approval from the authority and we have their advice, but still it's not a binding. So once we go for the quality, we'll go section, we finish the quality compatibility, we'll go to the authority, we have their approval, and go for the clinical. Since then we'll move to the clinical type. This is the way. Full dossier, of course. Now for a biological, we need to go for phase 1, phase 2, and phase 3 studies, clinical studies. Then approval. Usually, the patent is very long for a biological products. For example, in US, it's 12 years patent. In Europe, it's 10 years, and depends, of course, the authorities how it's handled. But we have a long time of patent of these products. While if we go for a biosimilar, it's the same approach, stepwise approach, accreditation from starting from cell bank and starting from early. Scientific advice are mandatory to have it all the time. But here we have to go for compatibility studies for the clinical to prove similarity with totality of evidence. We don't go for phase 2 studies. We go for phase 1 and phase 3 only. Phase 2 is not required for a biosimilar product in order to get approval. The same way, we are talking about stepwise approach. Please, I will have highlighted the proof of similarity and total of evidence. These two terminology are very important in addition to the compatibility studies. So, stepwise approach, we start with comparative analytical module 3 quality. If it is approved, we'll go for animal studies. And we can here, as I mentioned, we have waived some of the studies if this have been approved similarity. Clinical study, but still more studies can be required from the authority before approval. How to prove if comparability studies of module 3, the quality part. It's a side-by-side structure analysis, bioassays, we are talking characterization structure, and mechanistic differences between test and reference product. Usually we use the same reference products, but multiple of batches. About 10 batches of reference product should be used in order to waive the any differences between the products and to prove biosimilarity. And this is starting from the structure, mechanism, process, manufacturing process, early stages, the cell bank, the conditions, everything. If we prove this and we get a very similar product, will go for the animal studies. Here, toxicity and potentially some immunogenicity. As I mentioned, this might be waived to a certain extent if we prove this. Later, we need, for clinical, we need to go for pharmacokinetics products, complete pharmacokinetics, pharmacodynamic, and immunogenicity studies. Sometimes we start the immunogenicity study, since immunogenicity studies is a long type of study. we can submit, for example, 6-month immunogenicity studies and with commitment to follow up later after approval as a post-approval studies, and this is accepted by all the authorities. More studies, this is really depends on the assessment and might be post-approval studies. We have a conditional approval is given in order to practice use it in the market, and then we follow up the studies. This is the way how a biosimilar approach of the product. If I want to talk about what do we mean by totally of evidence, this is we need to understand it very well in order to prove our biosimilar products. As I mentioned, structural, functional analysis of reference product, propose, comparison, purity, chemical, identity, bioactivity. So it is a proof of evidence there is no differences in purities, no differences in chemical identity, no differences in bioactivity of the product. If we prove of these, this is a type of total of evidence. Minor differences is allowed are allowed if there is such for example, differences in stabilizers or buffer. This is accepted, but should be justified. No meaningful clinical differences. What do we mean? Refer to differences safety, purity, and potency. Demonstrated through human pharmacokinetics, exposure, and response studies. And of course, we are talking about patient, long-term studies, clinical studies in order to prove. Additionally, clinical studies might require other than immunogenicity. If a single study is submitted, it will not demonstrate biosimilarity. So single study is not approved. So multiple studies is needed. I want to show this is a product, a reference product, and a biosimilar product. It looks like it's the same. But if we go through a little bit more, we can find here a little bit change between the two products. If you can imagine, this was clinically inactive. This minor difference between the reference and proposed biosimilar product. This caused a clinically inactive product, biosimilar product, and it was, of course, withdrawn and recalled from the market. Now, the word of interchangeability with a reference product. This really have been a long debate with interchangeability. Can a biosimilar product be substituted by substitute the reference product? I remember I have attended maybe 2 or 3 conferences about interchangeability between biosimilars and reference product. Of course, it was very hard to a biological product manufacturers to accept biosimilars interchangeability, and it was very hard to the authorities to prove that. But at the end, there have been really very debate, and we have many really thoughts about that. But most of the countries accept the now recently accept the interchangeability in a way that you need to prove that more, and sometimes you have to submit more studies to prove that. So at the end, not all biosimilars are interchangeable. So interchangeability should be within the approval process. That means the authority will finish up that this biosimilar is classified as interchangeable with the reference biological product. You can't say that my biosimilar is interchangeable depending on your studies unless it's approved by the authority and classified as interchangeable. And of course, in order to avoid this, for the approved biosimilar that is not interchangeable, if you start, you can't start an old patient using a reference biological product to shift it to or substitute with a biosimilar. No, you can introduce a new patient on the on the biosimilar products, and you continue with this biosimilar product. This is the way with what we mean by interchangeability. For a biosimilar to be classified as insured with a biosimilar must meet additional requirement. This is what I mean. This is the additional requirement that, sorry, the authority asks for in order to prove the interchangeability. The manufacturer of interchangeable biosimilar must provide additional information showing that the product provides the same clinical efficacy and safety as the reference product, including the effects of switching back and forth. Switching back and forth with the reference product. The second concept of is the immunogenicity. Immunogenicity is the ability of foreign substance to enter a person's body and cause an immune response. This is very important, and since we are talking about biological products and we are talking about same proteins and antibodies, large complex carbohydrates, so there is immune response. Like all biological biosimilars, may elicit unwanted immune responses that can significantly impact the clinical efficacy and safety. So, head-to-head immunogenicity assessment of biosimilars and their reference biological should therefore be a critical component of biosimilar clinical development program. So, none of there is no biosimilar dossier that should be submitted without any immunogenicity studies. As I mentioned, recently, FDA have approved that for insulin, since we have, it has been introduced since 1980, and now the molecules have been in the market for more than 40 years. So it has been a little bit, it's not necessarily to submit the immunogenicity studies for NDA approval, but for MABs, for example, and for other products, no, we need to submit, and we have to refer to each product guidelines in order to submit the immunogenicity and the extent of the study. Either we need 6 months, 12 months, 2 years, depends on the molecule itself. How to reduce this immunogenicity? Immunogenicity usually happen by more therapeutic proteins. If we have a multidisciplinary risk-based analysis early in product development, in order to minimize immunogenicity, we need to take care of early development of the product. The higher the risk category for the product, the faster the pace of assay development should take place. There are many factors which influence immunogenicity responses to therapeutic proteins. It is a safety concern. There is a need to assess and measure it, correlate it with all clinical data, pharmacokinetic, pharmacodynamic, all of this should be correlated in order to minimize the risk of immunogenicity. There are regulatory expectations from the authority. Any authority will consider, provide risk assessment and appropriate sampling plan. For example, I started with this cell. I checked this cell. It's the cell bank was the master cell bank, but the working standard cell banks were identical. I have checked all this. I have the bioactivity of them or similar bioactivity, so I can proceed. I've done all the assays, the biological assays, the binding antibody assays, neutralizing antibody assays, and they were similar. So this will minimize the risk of immunogenicity. It is, of course, phase-dependent assay development. Have assay validated prior to testing. Always validate the assay, and it is a bioassay. We are talking about a bioassay before any clinical and any phase study, 3 studies sampling. Crucial to have appropriate stored study samples. Then always keep the retrieve data, the samples in order to recheck again. And of course, the storage condition of all this stuff is very critical, very critical. Any change in temperature, any change in humidity will affect the cells and will change the whole cells. The main difference between chemicals and biological, as we mentioned, refers to an active substance master file. We are talking about master file certificate suitability. This is not really applicable for a biological product. There is no certificate suitability. There is no active substance master file. You need to go for the cell line, the cell bank, how to prove it, and all this. API size of chemical product need only GMP to prove, but here no. You need to accredit the cell bank. You need to visit the place where the cells have been incubated, have been harvested for fermentation, all of this stuff. You need to accredit it before. Registration guidelines are mostly harmonized for generic product and chemicals, while it's not for biological. Cost of development is cheap and fast in comparison to biosimilars. Biotechnology is difficult and complex. This is our references that I have referred to, and I would really appreciate if you go through them and read them. Go for the FDA and each requirement. Jordanian FDA biosimilars and Egyptians are very detailed. You can really refer them as reference to understand more about biosimilars products. I would like to thank you all. I hope I have really highlighted the most differences or important between both of them, and I'm ready for your questions. Thank you.
Thank you, Dr. Hakima, for the very detailed, rich presentation. We really enjoyed our evening with you. I wish that this webinar would never ended. However, I would like really to know more about biologicals and biosimilars because they are really important medications in this era, as we had said. And you had just make this topic simple and really enjoyable to learn and to hear more about. Thank you very much. Now I will move to the Q&A because you don't have much time so to hear most of the questions. But before we move to the Q&A section, I would encourage everyone who had attended the webinar to register to our listserv. Doctor, do you mind if we share the slides with the audience after the webinar?
No, I don't mind.
Okay, so please register to our IVBN Regulatory Affairs listserv so you can receive the presentation and to see the slide, also to refer to the references that Dr. Hakima had advised you to read and know more about. Every time you read more, you can gain more knowledge, and every time to listen to any webinar, you can really connect and collect every details to get all details together. It's like a puzzle. You collect our information and then you will have a full image about the biologicals and biosimilars regulations. And this webinar was very informative since it had highlighted many different authorities like the Saudi FDA and the Jordanian FDA, also the Egyptian and many other authorities. So please register to receive the presentation. Also, this session is being recorded, so we will download the recording to IVBN Regulatory Affairs listserv on IVBN list website library, so you can refer to it whenever you want to. Also, we will post it on LinkedIn once it's available. Now we will move to the Q&A question. If there is anyone who's interested to open his mic and ask Dr. Hakima directly, please raise your hand so we can arrange the questions, and I will start looking to the Q&A section. Okay, we have the first question from Abdo. Are there any required tests like potency assay for secondary packaging products, biosimilars, like second labeling? What are the pharma companies doing now in Jordan?
Yeah. Are there any potency and no? So since you are talking about finished product, almost secondary package products, so the dossier will be coming from the licensor. So this is a contract manufacturing type of a product. The only thing that you will, for a secondary package, for example, in Jordan, you will be the MAH holder, so the marketing authorization holder. So you have to be a batch releaser. Unless you have, depending on they rely on the batch release of the original product. But in general, in Jordan, they will accept reliance on the marketing, on the manufacturer release or but in some countries, no, you need to go through, again, batch release. So after secondary packaging, you need to go for batch release. Other thing that if the secondary packaging contains any type, usually it is, of course, there is no heat or no direct contact or environmental condition with the secondary packaging, unless it's a functional one. If it is a functional secondary packaging, then it have to go through more details, and we might need stability if the secondary packaging is functional. So we need to understand what is the role of the secondary packaging, functional or non-functional. Thank you.
Following to this question, Abdu had asked if there's an acceptance criteria or there is a plus or minus difference in the percentage of the. Let me go back to the. When he asked about the potency assay, he is asking if there's an acceptance criteria or plus or minus difference in this test.
No, as I mentioned, we need to be similar as much as we can. Now, there is no range of, for example, 90 to 110, usually in the assay. But there is, of course, some ranges, to some extent, depends on the cell itself, the potency of the cell. But we are talking about potency, we are not talking about assay. So it's not a chemical product. Depends on the molecule itself and the header, but usually we need to be as much as close to the reference product.
Okay. Another question for locally manufacturers, if the local manufacturer is supposed to do only secondary packaging and batch releaser, do we consider it a local manufacturer or the product must be fully manufactured locally?
Depends on the authority. For example, in Jordan, this is accepted. But for example, in Tunisia, this is not accepted. You have to have more involvement in the manufacturing process.
Okay. Is the manufacturing process of biosimilar products must be exactly identical to the biological one? No meaningful difference.
Sorry?
Is the manufacturing process of biosimilar products must be exactly identical to the biological one? No meaningful difference.
Yes, as I mentioned, the product is the process. In biological product, the manufacturing process is the most important really for the biological product. Always keep in mind, the product is a process. If there is a change in the process, there is a change in the product. So it should be exactly the same.
Exactly the same. I thought there's another question. I thought for biosimilars, we need a new biological license and not a new NDA, right? Abdo is asking again.
It is a new product. It is a new product. It is an NDA or new drug applications. Let's consider it.
Okay.
Yeah.
Bioanalytical method for biosimilar test must be same that used in the biological license BLA 1, or slight deviation will be accepted and enough to be validated to validate the whole method.
Slight differences that have no effect on the assay, or for example, you are going to the state of art. We are talking about the state of art method of analysis. The biological product have been the reference, I mean, have been introduced and developed and manufactured 20 years ago. So we are talking about state of art. So there might be some differences, but we are talking about state of art. And the second, we have validated method. So we need to go for a validated method. Yes, sure.
Okay, we have same product, but we need definitely different batches due to the variability in the reference product itself, right? Someone is asking.
Again, please.
Same product, but we need definitely different batches due to the variability in the reference product itself.
True. True. That's why we go for more than one batch, as I mentioned, sometimes 10 batches or something like that, in order to minimize the variability in the reference product, because the reference product is a biological product also. So there might be some differences. So in order to minimize the risk of these differences, we'll go for more than one batch in order to have to make sure that we have catch up all the differences, little differences, if there is.
Okay. Dr. Hadil is asking, a state-of-art method of analysis means that the company develops its own one, right? Or
no. State of art should be published, known, there is a literature, there is differences. Unless they have this method and go for full validation of this method and prove that it is a state of art. Of course, you can innovate anything with this, but you are talking about biosynthesis, bioassays, so we are talking about now more than the infrared and the norm type of method of analysis. Yes, sure, we go.
Totally agree. Yeah. Yes. In the BLA was indicated for 3 or 4 indications, then is it accepted to prove the similarity of 1 or 2 indication and waiving the last?
As I mentioned before, it wasn't accepted to extrapolate for the indication, and you have to have head-to-head study, clinical study for each indication. New guidelines or new that have permitted some, if you have totally of evidence of the product, then you can extrapolate. Depends on the molecule, depends on the product itself, and depends on the scientific advice of the authority.
Okay. What does compatibility studies include in brief, please? They want a brief about what does the compatibility studies include?
Everything.
Not a brief, it's going to be detailed.
Detailed in yes. Each step, compatibility studies, quality, non-clinical, clinical. I'm talking about everything, yes.
Okay, everything.
Starting from the cell.
Okay. We have another question from Dr. Sara. Is the biosimilar has different shelf life than the originator? Does it mean that the biosimilar product failed in the comparative studies?
Usually it depends on the stability studies that you have conducted. We must acknowledge and know that the biosimilar products is a biological, and it is sensitive temperature product, sensitive to humidity. So, for example, if I have an EU approved product with the temperature of 25 degrees and the humidity and for zone 2, and I'm transferring the biosimilar to zone 3 studies, and I don't have enough data for zone 3 studies. The 5 degrees differential between 25 to 30 is a major difference in the biological product. And usually we have an extra conditions and extra precaution regarding the temperature changes. And so we need to fulfill that. Sometimes we have to shorten the shelf life in order to make sure it is stable. Yes.
Okay.
So that does not mean it's not fulfilling the comparability studies, but that does mean it is not fulfilling the complete stability conditions under the certain conditions.
Okay. Another question, how many of batches of biosimilar test products required for establishing biosimilarity?
Minimum of 10. This is what I go for, 10 batches or even more.
Okay. What stages of biosimilar batches are preclinical stage, clinical stage, and validation stage to be used for establishing biosimilarity?
What? The first.
What stages of biosimilar batches, preclinical stage, clinical stage, and validation stage to be used for establishing biosimilarity? Is it clear, or shall we ask for a clarification?
Yes, but I don't know. I have mentioned the stepwise approach. So we need to start from quality to non-clinical and we have to go comparability studies from quality to non-clinical, and maybe this is the answer, I think.
Okay. Please, if you have another clarification for your question, please type your question in the Q&A chat section. Okay, about interchangeability, is the announcement of interchangeability by the reference biologic product obligatory? Is it obligatory?
The interchangeability is for the biosimilar, not for the reference product. So the authority announced that approved the product to be interchangeable biosimilar product.
Okay. Since we need to be similar with head-to-head comparability, how does the biosimilar is considered cheaper than biological? This
is a good question. and always I'm really argue with this. You should have the same price because we are going to go for head-to-head studies, and but still the innovation, the known, the safety, the public, there you can rely on published data of the reference product. So no, it is cheaper now. That it's not that much cheap. There's a slide I was preparing how much the difference between the price of the reference product and the biosimilar, but I think it's out of the scope of our presentation, so that's I was deleted. So I have deleted, but it is less than 15 to 20%. Oh. Now, considering a generic product, you are talking about 35 to 30% less than the original product. So it's although it's cheaper, but it's not that much cheap as the generic product, chemical generic product.
Okay. They thought, I think, about the patients in this point.
You know how much millions have been really saved once we use the biosimilar products.
Yes,
because they are expensive. Even the biological, yes. The original is very expensive. Yes.
Especially in our region.
Yes, yes.
What impurities to be controlled in biosimilar product specifications, and on what basis of limits to be established for impurities which are not detected in reference product?
Still, I'm talking about impurities of, for example, amino acid, through glycosylation, through agglomeration of the amino acid, or something like this. We're not talking about chemical, for example, mercury or something. We're not talking about chemical product. We are talking about proteins. We are talking about biological. So there shouldn't be the purity profile. Impurities shouldn't be that much difference between the reference and the. Nevertheless, it's accepted if it is justified. And if you know that these impurities have nothing to do with safety and clinically or the efficacy of the product.
Okay.
So it is permitted to have a little bit, but you have to recognize what type of amino acid have been done. They shouldn't develop any immunogenicity studies. The most important thing is the immunogenicity, because any protein or an amino acid might might cause immunogenicity. In this case, we go for non-clinical equivalency between two products.
Different in the shelf life than the originator, considered as non-meaningful different, or must be exactly the same as originator?
I think we have answered this question.
What are the difference in stability studies between chemical products and biosimilars one?
The main difference is that you have their chemical assay, validated method, impurities are known or unknown according to their material. We have a pharmacopoeia. We have a monograph for the chemical. We can refer it, but for biological, we don't have anything of that. There is no monograph. There is no. We are talking about a bioassay. We have to have amino acids assay. We are talking about natural living cells assays and so it's totally different. And the methods of analysis, the methods are totally different between both of them.
Okay. I'm just trying to skip the questions that we had answered to focus on more point. If the biosimilar product have the stability studies cover the whole requested shelf life, does it will be accepted? Would it be accepted?
Yeah. The answer is yes. If you have full shelf life, usually the accelerated. I have to mention here, accelerated studies in early development is very important. Why? Okay. In order to see the impact of temperature on the product, because temperature is very, but later you don't need any accelerated, because once you developed the how much the impact of temperature, you can go for the product, since you know temperature, as I mentioned, that the change in the egg while boiling from egg to boiled egg. So this is the impact of temperature on the protein. So you don't want that change, of course, right, in a biological product. What we end up is that we do the accelerated in early stages of the product once we raise the temperature to 40 degrees centigrade, and we are sure that up to 40 there will be a changes. But by this case, we understand what the reflect of the impact of the temperature on the product. Later, we don't go for further such accelerated studies, because we understand how the difference between the reference and the biosimilar products through temperature accelerated stability studies.
Okay, I had imagined boiled medical.
How the results of a biosimilar.
And chemical drugs, we refer to pharmacopoeias to set specification limits. Is there any pharmacopoeias for biologics or biologicals or biosimilars? Is there? No.
The answer is no, as I mentioned, no monographs.
Only for chemicals.
Yes.
If not, how we set the limits?
Now there is a published data for each product. For example, insulin have.
Okay.
I don't want to mention it is a monograph because it's not a typical monograph, but there is well-known data, accumulated data through the 40 years of biosimilar information about the product. So you can refer to such published data.
But I think because insulin is known as a molecule since long time ago, maybe yes, it's like a similar monograph, but when it's come to new biologicals, or it's not the same case.
Yes, sure.
Do we need a freeze the stability for biological and biosimilar products intended to be used IV?
The question, I think, about the freezing, thawing, stability studies.
Yes.
Usually, most of the biological products are refrigerated because they are temperature sensitive. This is what I've mentioned, Hada. They are all refrigerated. So freezing and thawing is part of this stability studies in order to also understand the effect of lower temperature on the product, because we are talking about protein. Now, usually, minimum, the effect is not that the highest as the same as the high temperature, but still we need to do it. Freezing, thawing, yes, sure. And usually many multiples of cycles of freezing and thawing. Yeah.
Okay. Do we need comparative stability studies to compare the biosimilar to BLA? If yes, then how many batches needed to do that? And what are the tests to be included?
As much as you can. And you have to start from the cell itself. So we need stability for, if you mentioned drug substance and drug product, finished product. Stability, compatibility, stability studies. You have to show this in your dossier. We need to refer, once we assess, not we, the authority assess the dossier, need to see all these stepwise and see the comparison between the two products. Yes.
Okay. For biosimilar to be considered as interchangeable, the manufacturer must submit additional information. Is this a request submitted by by manufacturer as a special request, or it will be with the registration process by default?
Usually, the aim of any biosimilar manufacturers to have an interchangeable product. So through scientific advice and with pre-meetings with the authorities, I prefer to go through, and I want my product to be interchangeable. What's the extra studies that please advise what to do in order to reach to such interchangeability? So it is the objective of any manufacturer to have an interchangeable biosimilar. But the results of this interchangeability depends on the authority by itself. Now, of course, there are some physician and let's say it is they considered it as interchangeable without referring to the approval of the authority. There is such a use, I mean, or misuse or whatever, but usually if you want to be registered as interchangeable, you need to ask the authority what extra studies you need in order to have an interchangeable product.
Okay.
And depends on the number of amino acid in your molecule. For large molecules, it's very difficult. For small molecules like insulin, it's possible.
Yes. So that this was the advice from the expert. Okay, can we replace insulin that has the same active substance? Can we replace insulin that has the same active substance?
Still, this is really, go back for the previous question. It's the same. Insulin, to some extent, is from 40 to 100 amino acids, so it's considered a small biological product. Small. So the interchangeability and the biosimilarity switching regulations are much less simplified or more relaxed than other, but still you need approval.
Okay, we want to take one final question for the sake of your time, Dr. Hakima. Can we safely interchanging insulin with the same active ingredient, different trade name?
The same answer, if it is, yes, the same question as before, yeah.
Okay, those was the full questions from the Q&A section. Does anyone want to ask a final question for Dr. Hakima? Please raise your hand because we have only 5 minutes left for our webinar. Unfortunately, only 5 minutes left. Okay, Malak, please. Malak, can you hear us?
Maybe she's mute.
Yes, she's muted. Malak, you're muted. We can't hear you. Okay, in the meanwhile, we would really encourage everyone to register to our listserv so you can receive the presentation and receive all the announcements on the upcoming events. Also, if you have any suggestions on any topic that you really want to hear or to know about, please share your thoughts with us via our listserv. There's Adel, Dr. Adel. Dr. Adel, do you want to ask Dr. Hakima? Dr. Youssef, can you please unmute yourself and ask Dr. Hakima? They are muted. I don't know if they can hear us or what. Therefore, would you please check if the audience can unmute their selves and speak? Dr. Youssef is with us, I think. Dr. Youssef, can you hear us?
تحكي معي؟
Yes. Can you ask Dr. Hakima because we have only 5 minutes.
أنا إنجليزياتي ضعاف ما أنا مش فاهم.
تفضل in Arabic تفضل.
اللي بدك إياه.
شهادة شو اسمه التصنيع GMP هاي. يعني بكل واحد مثلا يصنع في دولة ويكمل تصنيعه في الأردن مثلا.
Yes طبعا إذا في contract between the two companies sure بقدر.
ما في، أنا بدي أجيب أصناف وأتفق مع شركة أردنية يصير يكملوا في الأردن، بيصير إنه مصنع برا ونكمله في الأردن.
مصنع according to GMP بمصنع.
مع شهادة التصنيع MG يكونها.
شوف إذا معك شهادة جودة تصنيع وإذا أنت مصنع بمصنع أو أتليست عامل، المهم تكون عامل بايلوت باتشز
بس
اللي
هو
هي تشغيلات تجريبية. إذا عندك تشغيلات تجريبية وعامل optimization and of the formula وعامل full development of the هذا you can make a contract manufacturing مع أي شركة أردنية أو غير أردنية. This is what's called technology transfer. وتكمل تصنيع. شرط انك تكون انت اول إشي او اتليست بدك تبيع الديفلوبمنت تبعك. ممكن تكون انت بس بدك تبيع الديفلوبمنت مش تعمل كونتراكت مانيفاكتشرنج، يعني بتبيع الاوبتمايزد فورمولا اللي عاملها انت بتبيعها للشركه وهي الشركه بتكمل.
هو ما بينباع في دولة المنشأ.
ما في مشكله.
ما في مشكلة عادي يعني.
بيصير لوكالي مانيفاكتشر بهديك الدولة ما هو الثانية.
اوكي. شكرا لك دكتورة.
أهلا
وسهلا.
بس هذا out of البيوسيميلار.
Anyone wants to ask because we have only 1 minute? Okay, at the end of our webinar, Dr. Hakima, we would like to thank you really for accepting our invitation and joining our learning initiative for tonight. We had really enjoyed every single moment from today's webinar, and we had really learned a lot from you. We are really looking forward many webinars, inshallah, if there's any other opportunities. Our audience, we would like to thank you all for being with us tonight. Really, you were amazing. The questions were amazing, and you had really added a lot to our webinar. We were pleased to have you all tonight. We have a big audience for tonight. I'm very happy because a lot of people had the chance to really attend. We would like to thank all the IVBN Regulatory Affairs working team, Dr. Muna, our leader, Dr. Muna Al-Musli. Thank you for your continued support and thank you for everything. Rafa, Kulthum, thank you for everything. Also, we need to thank our colleagues at IVBN, Dr. Hussam and Dr. Ahmad and all the IVBN family for helping us and supporting us to have a successful webinar for tonight. We look forward to see you in our upcoming webinars. Dr. Hakima, we wish you a very great day ahead since you're based in Canada, and we wish you a good evening for every one of you. We will see you inshallah next time, and don't forget to register in our IVBN listserv so you can receive the presentation and learn more and more from all the learning material. Thank you, Dr. Hakima.
Thank you very much for everything. Really, I enjoyed this session. I was really hoping that I have given something new and clear for everybody. I wish to see you again, inshallah. I can really share my Hotmail if anybody wants some questions regarding biosimilars. I will share the presentation lately, inshallah, but after I finish, I'll come back. So thank you, thank you, IB and everybody in this. Thank you, Dr. Mona and Rafa, Dr. Ahmed, and everybody really for helping us. And thank you, audience. Really, really, I appreciate it. I'm very proud. I have such type of people that are really very knowledgeable and looking forward to understand and hope we'll see each other again. Sure.
And we are very, very proud of you, Dr. Hakima. Thank you very much. Thank you. You're such a role model. Thank you very much.
Thank you.
And best of luck.
Thank you. You too also.
Good night, everyone.
Good night.
Good night, everyone. Bye-bye. See you. Bye-bye.
Bye. Assalamu alaikum.
Alaikum assalam. Ma salama.